Hello friends, today in this video I am going to tell you about top 10 interview questions and their answers. Everyone is prepared for interview, either you are fresher or you are experienced. But whenever we prepare for interview, we want to know about the questions and answers because interviewer asks the question, we want to give satisfactory answers to that questions. And that's why here I prepare this video to discuss that top 10 interview questions and answers. So let's start. So these are the 10 important questions which are asked in interview. First question is, what are the different types of tablets? Second, what is theoretical plates? Third, what is the principle of UV spectrophotometer? Fourth, which are the components of HPLC? Fifth, what is the column? Sixth, which are different types of column used in HPLC? 7. What is Isocratic system for HPLC? 8. What is gradient system in HPLC? 9. What is normal phase HPLC? And the last question is, what is reverse phase HPLC, which is also called as RPHPLC. So these are the top 10 interview questions asked in interview. So let's see the answers of these particular questions. First question is, what are the different types of tablet? This question is a very important question. Okay, so the first thing, you should define the tablet. So the tablet is defined as a compressed unit solid dosage form containing medicaments with or without excipients. Now enlist the types of tablets. So here I have mentioned 8 types of tablet. First is a compressed tablet. Second, multiple compressed tablet. Third, delayed release tablet. 4, sugar coated tablet. 5th, buccal tablet. 6, sublingual tablets. 7, film coated tablet. And 8, effervescent tablet. So these are the different types of tablets. Now the next question is, what is theoretical plates? Now theoretical plates, this concept is related to HPLC. Okay, so you can define the theoretical plates like this. Theoretical plate is a measuring tool of HPLC column efficiency. Any chromatography column does not have any physical plate, but it is a result of mathematical calculations, and the column having a high number of theoretical plates are considered as more efficient HPLC separation. So you have to answer like this to this question. Third question is, what is the principle of UV spectrophotometer? Now UV spectrophotometer is the instrument which is used in pharmaceutical industries. Okay, so you have to know the principle of this UV spectrophotometer. The principle of UV spectrophotometer is based on the absorption of ultraviolet light or visible light by chemical compounds, which results in the production of distinct spectra. Spectroscopy is based on the interaction between light and matter, and when the matter absorbs the light, it undergoes excitation, and the excitation resulting in the production of a spectrum. So this is the answer for this question. Now next question is, which are the components of HPLC? So here are the 6 important components of HPLC. First, pump. Second, solvent reservoir. Third, injector. Fourth, column. Fifth, detector. And sixth one is a recorder. Next question is, what is column? Now again I want to tell you about column. Column is related to HPLC. HPLC, that is high performance liquid chromatography. So the column are main components in HPLC because the column is responsible for the separation of sample components. Now this column is consist of stainless steel tube with stationary phase. Now, which are the different types of column used in HPLC? So, there are different types of columns are available for HPLC, and the selection of column is depend upon the sample. Okay, so here I have mentioned three different types of HPLC column. First is a C4, second is a C8, and third one is a C18. So these are the main important column of HPLC. Next question is, what is isocratic system for HPLC? So isocratic system for HPLC is depend upon the composition of mobile phase. Now when the system in which the mobile phase is constant in runtime, that system is called as a isocratic system. Okay, in isocratic system, mobile phase is the same all over the analysis. Means there is no change in mobile phase composition, and hence this system is called as a isocratic system for HPLC. Next question is, what is gradient system in HPLC? Gradient system in HPLC is the opposite system of isocratic system, because in isocratic system there is a use of single mobile phase during the runtime, but in gradient system in HPLC there is a composition of mobile phase is varies during the runtime, and hence this system is called as gradient system of HPLC. Now, what is normal phase HPLC? In normal phase HPLC, the stationary phase like silica gel is polar in nature and mobile phase is non-polar in nature, and in this technique non-polar compounds travel faster and elute first. This is because of less affinity between the solute and stationary phase. Okay, and this phase is called as a normal phase HPLC. Now next is a, what is reverse phase HPLC, means RP HPLC. So in reverse phase HPLC technique, a non-polar stationary phase is used and the mobile phase is polar in nature. Hence, polar components get eluted first and non-polar components are retained for longer time. Since most of the drugs and pharmaceuticals are polar in nature, they are not retained for longer time and eluted faster, which is advantageous in this field. Thank you for watching this video. Please subscribe my channel and best luck for your interview. Thank you.